Welcome to Infectious Disease Lesson 1.3, What is an Infectious Agent? Part 1, What Bacteria Need to Make Us Sick. The goals of this lesson are to convey how our daily habits minimize exposure to pathogens and to introduce bacterial structures and the roles they play in infection. At the end of the lesson, the students should be able to explain common practices used to avoid infectious diseases and describe the bacterial structures that are critical for their roles in infectious disease. We will achieve our goals by brainstorming everyday life practices that prevent transmission of infectious diseases, drawing a bacterial cell as a group in order to familiarize ourselves with bacteria, and engaging in a jigsaw activity on bacterial structures and functions. To prepare for this lesson, you'll need to review the following key scientific concepts. Some bacterial structures are similar to eukaryotic cell structures, while others are specific to bacteria. And bacterial structures have a function and a role in survival and infection. You can review this scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure. And the student workbook provides additional explanation for students. Be sure to print the in-class worksheets, the jigsaw readings, and the homework worksheet. The key point of the Do Now is that daily habits play an important role in prevention of infectious disease transmission. We'll get there by brainstorming using homework responses. The Do Now slide begins by having students use their homework to brainstorm a list of behaviors that we all do every day to avoid spreading infectious diseases. This sets the stage for a later discussion of how infection can be transmitted. The key point of the lesson's activity is that many of the structures prokaryotic cells or bacteria use to sustain life are similar to structures found in eukaryotic cells. We'll get there by using prior knowledge of eukaryotic cell structures to draw structures that a prokaryote might need. To begin the first activity in this lesson, have students use their prior knowledge of eukaryotic cell structures to predict and draw the structures that a prokaryote might need. For example, students may expect a prokaryote to need structures like DNA or a cell membrane. Students may struggle, but the point is to prime students for the upcoming jigsaw activity, which they will use to add more structures specific to bacteria to their drawing. The key point of the lesson's second activity is to demonstrate that bacterial cell structures have a function in infection as well as in survival. We'll get there by completing a jigsaw activity on bacterial structures. For the jigsaw activity, have your students work in small groups to use the reading to complete the table in the worksheet. The jigsaw works best if each group focuses on one structure. In this jigsaw, we will learn about 8 cell structures that are specific for prokaryotic cells. The capsule, the gram-positive cell wall, the gram-negative cell wall, flagella, pili and spores, and nucleoid and plasmids. The success of the jigsaw activity depends on the reading, so you'll want to be sure to check that the jigsaw readings are at an appropriate reading level for your students. And you might want to assign the reading in advance if you think your students will need extra time to complete it. Once the groups have completed the table, it's time for a teach back. This can be done by rotating students so that each table has an expert on every structure, or by a whole class discussion. When discussing the structures, it's important to emphasize the functions of each structure in bacterial survival and infection. Note that some structures are found in all bacteria, while others with specialized functions are limited to select types of bacteria. The capsule protects the cell from environmental challenges and camouflages the bacterium from the immune system. The flagellum enables bacteria to move actively and quickly to new locations to locate a more favorable environment. All bacteria have a cell wall, which may be either gram-positive or gram-negative. We will go into how we can tell which in a later lesson. As we might expect, the cell wall protects the internal structures from the environment. The cell wall can benefit the host by causing inflammation, which enables the immune system to identify and eliminate the bacterial infection. The cell wall is also the target for a number of antibiotics. Some bacteria have specialized enzymes in their cell wall that allow them to resist antibiotics. Pili act like Velcro, enabling bacteria to stick either to each other or to structures in the host, like the epithelium in the nose. If the environment is not conducive to survival, some bacteria can form spores, allowing them to stay in a dormant state like a seed until the environment becomes more favorable. Like all living things, bacteria have genetic information in the form of DNA that encodes the proteins that make up its structures. Unlike eukaryotic cells, bacteria don't keep their DNA in a nucleus, which allows them to replicate faster than eukaryotes. Also, unlike eukaryotes, bacteria can also carry genes on circular pieces of DNA called plasmids. They can then transfer these plasmids among other bacteria. To learn more about each structure and its functions, review the key points in the worksheet answer key. The key point of the lesson's wrap-up is that structure determines function. To emphasize this point, we have included a graphic that most students find interesting. The key point of the lesson's homework is that bacterial structures can play a role in infection. We'll get there by completing the homework worksheet, which prompts students to relate bacterial structures to different infection scenarios. One of the most common questions students have is, can antibiotics treat viral infections? No, antibiotics target structures or enzymes that are present in bacteria that are absent in viruses. At the end of the lesson, collect the lesson worksheets to assess each student's table from the jigsaw activity. The bacterial structures covered in this lesson will be used when we learn about specific pathogens in Units 2 through 4. The concept that structures have functions will be further explored in the next 2 lessons. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.